There
screen.
it is, yeah. Side there. I think it's the one with the red bar. But
okay, so one of our coral biologists is advocating that we collect a sponge when we can. So to give him a little bit of coral love, why don't we look at this fish for a bit and then focus on this orange whip coral, and then we can grab a sponge if possible, please.
Okay, I'll stop the ship.
Chris, these are the same sponges which you... Chris, these are the same sponges you were looking at earlier?
Right.
What type of fish was this, Bruce?
Okay, it looks like another Antigonia, but this has a totally different color than the others, and it's really unusual. I don't think I've seen anything like this before with the sort of golden yellow and the grayish bar. Usually the bar is bright red and the body's sort of a silvery color. I don't know if this is just a behavioral color change or something, or if it's actually a different species, but the color is highly unusual one.
So these are the boarfishes, Bruce?
Yes, it is another boarfish.
Yeah. Sure. It's interesting, the boarfishes, catfishes, dogfishes.
It's always good to equate it to something you know.
Meow. Yeah, meow.
But we just saw one of those boarfish, what, about 20 minutes ago? And it was
totally
that typical color. It
no, it was totally different color.
The one before this
yes,
one. Yeah.
Exactly.
Oh, sorry, yeah, full white.
Sorry, I thought you meant it was...
No. The one he was describing from about 20 minutes ago was the one that was the white with the red. And this one has that golden cast that looks very, very different.
So you want to take a look at this coral and collect the sponge?
Please, if possible.
Okay.
Chris, are you happy with that? Collecting one of these sponges and taking a closer look at this coral. Scott, pay attention to the coral, because if it's of interest, we can also do a quick collection of that, hey? If not, we can keep moving. We've still got an hour to go.
I am totally happy with collecting one of those sponges, believe me.
Okay, great. We'll do that then.
Sorry, Pilot.
Zoom, please.
Yep.
Seems just a sponge is all we need. Alright, we'll find one of the sponges that are facing towards us. Let's see if we can't get a really good shot of it. We'll collect anything here. Current's really looking now. Seems like until I lift off, it pushes me back hard. Okay, you can come on in. Alright, video, you can zoom. I'm going to try to get one of these sponge for them and get a really good look.
Copy that.
May not be the one we actually collect, but it'll be very similar.
Full zoom.
Thanks. So you want one of these sponge here, or do we want to wait till we have a coral sponge combo?
No, I think let's just do this one here for now, please.
Okay.
All right, let me spin up HPU.
So we just saw your question in the chat room, Diva. Yeah, I think collecting a couple more samples would certainly be worthwhile, and we're not restricted here, and we've covered a lot of ground today and got a lot of close-ups. So a piece of the sponge and a piece of that whip coral would whip icidid would be terrific.
Oh. Okay.
When they die, Did you hear that comment?
Could we get a partial zoom on?
Chris, we did hear that. Diva just went to go check with Kelly to make sure that she'd be okay with collecting a few more samples.
Great, thank you.
Yeah. For those of you who aren't aware, although I think probably everybody is, up until this expedition, when we sampled, we were allowed to collect 2 biological samples and 2 geological samples. That's been relaxed a little bit for this cruise, as well as being able to collect biological samples of dominant species. But I'm still not entirely sure exactly how many biological samples we are allowed to get. So Diva will find out and get back to us.
Watch your right toe. What's that?
Okay. So we want both, right? Sponge and coral, is that? Oh, we're waiting to hear back.
We're waiting to hear back right now.
Okay.
So I think we all agree that the sponge we're picking up, but we're waiting to hear back on whether we can also take a clipping of that other.
Oh, okay. So get the sponge.
Whip.
Get the sponge done.
Why don't if you could wait 2 more minutes, that would be great.
Okay. I can wait 2 more minutes. I'll just get all lined up.
It's a fair amount of downvert to stay put.
Yeah, it's pushing pretty hard. I have to keep 30 ahead just to not get blown backwards. So I'm putting that and then putting a little extra downvert just in case. but I'm 50 and 30, so that's pretty reasonable. Top side phase currents at 13 amps. Should we maybe drop a target and fly around and look at things, or just wait?
Give me 1 more minute. I'll go run after them. Hold on a second. Oh, yep. Give me 1 second.
Okay.
Okay, so... Oh, Chris, can you hear me? Okay, so scientists ashore, we would like to make clear that the sponge will be sample number 5 and number 3 for biology. And if we were to do a coral after, that would be number 4 for biology and sample number 6. So are either of those remarkable? And they first warrant going over our routine 4 samples a dive. And if you had to choose between the 2, which would it be? Or are they both remarkable enough to collect them? Because of course we are not at our end waypoint yet, and we could go further. Silence. Chris? Scott? Anybody?
We were just having a little discussion here. I couldn't believe my ears, but Les said that the sponge might be more important.
Okay, well, if Les, the coral expert, is saying the sponge is more important, and yes, Scott, I know everything we're seeing is remarkable. This is the problem of exploring a pretty much unknown area. Most of these species we're seeing are likely to be new or new records, and so everything is remarkable here, and having to choose between 4 and 6 samples, which includes 2 to 3 rocks, is always difficult. So I think there's a general consensus from Scott and Chris and Les that the sponge is more critical, so we're just going to collect the sponge, please, ROV pilots.
I like what you rolled that right into them.
After all that deliberation, we apologize, coral people.
We get a partial zoom,
Tomorrow, well, I don't know how much you'll see. No, you'll probably see quite a lot on the trench walls. But we will make up for this with some more coral collections. But don't forget, you have your Desmophyllum cup coral from earlier, as well as the Scleractinian, which was pretty brittle, and they did a remarkable job collecting it earlier.
So see this already knocked over one, but I just picked that guy up since he's already fallen over, or do you want me to get one that's standing up, just in case that one's not alive or something?
Was that to me, pilot?
Yep.
Sorry, could you repeat that? I didn't hear properly.
See how there's one on the ground that's sort of knocked over, and then there's a couple
Whereabouts in the screen?
Bottom of the screen, right there. It looks like a really nice big array. But,
Yeah.
I think it's laying on the ground, not standing.
I don't think it is. I think it's standing up.
Oh, okay.
Let's just go with that one. Yeah, that's fine.
Right. Sounds good.
Can we zoom? Oh wait. So before we collect, can we zoom on it and get some really good imagery, please?
Of course. Come on in.
And also put the lasers on for a second so you can get a scale.
See, it's broken at the bottom there, I think.
Okay, and then can we pan up to the one that was just past it to see if they look different?
Yep.
I know they do look quite different. Oh, well spotted, pilot.
What?
Can you zoom to one of the other ones so we can see if they look different? If whether the one that you're pointing out is actually dead.
Zoom in here. I don't think it's dead. I think it's just. Oh, I mean, it could be dead, but there's another one.
Let's just go with one of the other ones in case.
Okay. All right. I was hoping. Okay.
Pilots are probably going to do this, but it'd be great if they could get the whole colony if it's not that big.
Okay, did you hear that, pilot?
I will do my best. The hand is big and clunky, and these are short, so I will. That's why I was petitioning to grab the one off the ground. It would be more likely a whole specimen.
Chris, do you think the one on the ground will give you tissue? I'm concerned that it's been dead long enough to not.
I'll zoom again if you would like.
Yeah, sure. Let's get that one again. So Chris can come
Zoom
on
in? Yeah.
Chris, they're about 10 centimeters apart. Do you think this one will yield tissue for any molecular analyses? It is knocked over and separated from the base. Okay, just to be safe.
Sorry, could you repeat, please?
So, Chris, we're slightly concerned because this coral we're focusing on right now has been detached at the base. And so I'm concerned that it's been dead for quite a while and may not yield any tissue that would be good enough for molecular work. So, would you prefer us to get another one, which is still connected and obviously alive, or is this one sufficient? Because this one, the RV pilot Jeff thinks he can collect in one piece, but he's concerned he may not be able to do that for the other ones. But the other ones may be alive. Those are the two issues.
So dead sponges are a bad collection because not only is the tissue a problem, but also they lose some of the smaller spicules after they die. So a live one would be better. Basically, he doesn't necessarily have to get the whole colony out. It's just sort of stating that it might be easier to try to grab the whole thing and get as much as he could rather than trying to pick off a little branch.
Okay, great. So then, Jeff, let's get one of the other ones, please.
All right, we'll get a good zoom on it and pick it up. All right, continue on. All right, let's come in on this one, Talia. Come on in.
Okay.
There's one.
This one?
Yep. Does that one look good?
That's great, thank you.
All right. Oh, it's right between my toes. All right. Does Chris have any idea what the consistency? Are these glass sponge? Or are they squishy? Or are they?
I'm not sure. Chris, did you get that? Are these going to be glassy? So these are hexactinellids or cladorhizids? I mean, they are cladorhizids, sorry. Are they hexactinellids or demosponges? Okay, if you give me 1 second, I can find that out for you, Jeff.
Okay. Let's zoom on one of these. Okay. All right. Why don't you zoom out here then, where it's a little less clunky with the arm. We'll grab one of these. Give me a partial zoom.
Okay, so the demo sponges, so they should be a little bit more supple,
Okay.
but still just, I guess, be as careful as you can be.
All right. We'll reach out a little bit from the vehicle. It'll be a little more dexterous, and we'll be able to grab something. So let's go for this guy here. Go ahead and zoom video. That's a little small. There's so many, it's hard to pick one, huh?
Yeah, we're leaving it at your discretion.
That guy.
Maybe do the one we
Yeah.
filmed, yeah.
By himself.
All right, that's the one. We're going for it. Come out a little bit. Yeah, right there. Perfect. And, let's try like this.
Nav, do we have any free boxes, bio boxes?
Ooh, they look brittle, don't they?
I think the 2 outer bio boxes should both be open.
Mm-hmm.
Okay.
Oh yeah, it's crunched right away. That's not very simple.
Okay, the consensus here is that it might be stelastrid coral, the way it's real brittle like that.
Which is absolutely hilarious because we just debated about collecting the sponge or the coral. But hey, so it's like the best of both worlds, right? Well, maybe not for the sponge people.
So what?
So no, we're collecting. Go ahead. Don't.
Still want a sponge?
Yes. Well,
Okay. If we
which is
could-
now a coral, but yeah,
Oh,
we
it's a
still want it.
coral.
Yeah.
Oh, okay. Then is that laying down one's still no longer?
I still wouldn't get the one lying down just in case there's no tissue.
I have an idea. Since it's brittle, we don't have to cut.
So I think given that if it's really hard to collect a live one, I imagine just some branches would still be helpful if that would be easier.
Still some branches is good.
Anything that you can manage would be good. Thank you. That was not meant in a patronizing way at all.
I didn't take it that way. You can come drive if you want. No, actually you can't, but.
Would you like some zoom pad?
It's okay. Oh, I thought I could pluck it. They're just so small. Goodness. Do you guys have any suggestions? You're awfully quiet. Copilot, pilot, or Nav?
No, it sounds, looks good to me. I think the only thing is if you do pluck it, I think you'll want to close the jaws pretty quick or the kern will rip it out.
I wasn't keeping the Twitch right around.
That's
Yeah,
tough.
He's on there. So I do have to snip. Just trying not to have to snip.
If you grab it, we could put it in port. Should be a little bit easier to. Looks like it might work.
Just so low to the ground to get my fingers that low and to grab is just gotta set the arm down and drag the fingers along. Let's think about this for a second. Trying to pluck it just tears the branches off. It doesn't do anything. Trying to cut it while it's getting the iron.
If you
Look at this guy. What's that guy?
if you swing out a little bit more, maybe you might have a better angle to kind of lay it down.
This looked like an angle lined up with the arm, right?
Oh, there you go.
Yeah. Just
It should get some.
physics is just
Can
the geometry is just such that I can't. No.
I think you're gonna miss it at that angle.
Yeah, see. Geometry just isn't there on that guy. Sorry, Watch League. Given the size of these guys, the geometry, I'm just having trouble because of the size of the hand and the fingers and the length of the fingers. Grabbing a coral without touching the ground and first is difficult. We're just looking for one that's sort of perched a little bit. Here's a suggestion. Is this guy that's fallen over, where'd he go? Where'd the one that fall over? Did we lose track of him? No. Can you come partial? Oh, there he is. He's over on the left. Is it worth trying to grab one that fell over? Just to have in the box?
I think as we're getting low on time, yeah, let's just get one that's fallen over and
Coming out video? Sorry. Come in a little bit.
okay, sorry. So people are chiming in differently. So if collecting one that's alive of these small cell asteroids is too difficult, then just snip the end of the large bamboo next to you, and we'll call it a day, sampling wise.
Okay, we can also try to find one of these perched more as we're traveling.
I think because we still have quite a bit of time. Wait, what am I talking about? I think as we still have quite a bit of ground to cover in very little time,
Yep.
we should just snip a sample and go.
Okay.
Thank you, though, pilot.
Yeah.
Which side do you want them in, pilot?
Um,
Port or starboard?
it'll be easy drop, so we can do the port side outer.
Copy. Extending the port bio pin.
Get the yarn out of the way. Okay.
And ready to open.
Yep. Go ahead.
Just to confirm, we're putting this into the port outboard. Outboard.
Outboard.
Yeah.
Oh, box is closed.
All right, I'm going to closing the drawer. Retracting the pin, then extending. Okay, now I'm opening the drawer again.
Much better.
Okay, closing drawer. Okay. Retracting pen.
Great. Got a target? Dropped. Yeah, okay. Let's skedaddle, huh?
All right, I'm going to spin down the HPU.
Thanks.
Okay, great work. Thank you, pilots. Yeah.
So as you can see, we are still moving along that now very flat, low slope carbonate pavement. I expect, although I can always be surprised, I expect that this is what we will see all the way to our end waypoint.
Video switching out.
Cup
Hello, pilot. Show me the great video. Like, what is that little dark spot?
Which little dark spot? This guy?
Yeah.
That stock crinoid.
Yep. ROV lead, you are right. Same ones we've been looking at before with the gazillion arms.
This is really cool when we first found it. It's really, never seen one before today.
You got plenty of time before I get to you.
Yeah. Oh.
And again, this community is just so different to what we were seeing before. These very small
Did you get super close to
celastoid
it?
corals, which we previously thought were
For now,
sponges,
that's the end of my tether.
and a couple crinoids
Each in as I had room.
just splashed in between.
Let's get its legs.
You can also see a cup coral in the back. Desmophyllid of some kind in the upper left corner.
Up, down. Or set down.
Yeah, I'm tugging you.
Let's tilt down to the feet
Down to the feet.
at the right where it touches the ground.
And so you can see these very distinct long cirri that I think Chris was discussing earlier, which help to anchor it in place.
You can just keep going up.
Okay guys, this is great. Whenever you'd like to move ahead, we are happy to do so.
I think they're just waiting for Sirius to catch up.
Yeah, we had to get the ship moving after last collection, so we're skipping our time a little bit till we get caught up. Skipping our time a little bit till we get caught up. And we're done. Got a little tether.
Get quite a bit of shadow from the arm. I don't know if you guys can adjust the arm or the light bars.
Yeah, do you want to swing the
Uppers?
The lowers? Starboard lowers turn on.
Copy.
Make sure you get a good view of it on one of the cameras before you start moving it around.
Will do. So starboard lowers almost right up flat. So then,
What I would
So
like
just
from
make
this
sure
angle,
that
what I would like from this angle is since you're in flat, if you're comfortable with it, actually bring them all the way down, so we can light
so
more ahead.
before you do that,
yep,
take the lower swing arm and make sure it's turned all the way right.
copy.
If it's the starboard one you're moving,
Yep.
and that way you know that it's not going to swing into the craft. And watch it as it goes and make sure that it doesn't swing into any of the
Copy.
manipulator or
Moving
anything
right.
else.
Looks like it is all the way right.
Perfect. They use that one when they do white balance. It's really important to check, especially that one.
Okay. All right. So I'm deploying now.
And you can watch on the rail cam too as it goes.
Yeah.
Either one of those.
That's what I'm doing. About there. About horizontal.
Yeah. Another video says it looks good.
How's that look, video?
I think bring it maybe full back. We've got just a lot of light reflecting off very bright rock.
It already is full back.
Okay. And it doesn't rotate, does it?
No, you can't
No.
rotate up and down.
I mean, this has definitely got rid of the shadows at least.
It'll like karate chops towards the manipulator. I think it's got to stop before it gets there. But that would bring more light towards the center where you're saying it's hot. So I don't think that's what you want.
I think now my goal is to spread more light out
Yeah.
further.
Yep, and he's set for that.
Okay. Yeah, if we could just do that with both lights.
Okay. Yeah, it looks like the port is. That's strange. In
This
Sirius
is-
camera, it doesn't look like the port's deployed
This
at
is
all.
such a huge contrast to yesterday. We started with
It's this white moving thing at the bottom.
A barren
Crab.
volcanic landscape and ended up in a volcanic basalt. Yeah, yeah.
Because it wasn't entire. I was laughing because
so
it wasn't
with the
entirely
lower-
barren.
Right.
It was when
Well,
we hit that mix of
more
carbonate
barren.
and volcanics that it really became
With the port lower,
And
really
then-
barren.
And then
I swing it
now,
left
and then we
first,
moved
right?
into a very species-rich
Right.
and abundant area for biology in a carbonate area towards the end of our dive
Yeah.
yesterday. Whereas at the end of
Okay,
this
so
dive, we've
20.
ended in this
Got a
wave-cut
good
sort
view
of
on Sirius
flat.
camera.
It's not wave-cut, but
Maybe. We don't know.
this flat carbonate platform
Ready, pilot?
With not a lot going on.
with not a lot going on
How far do we
biology-wise.
have left?
It's really
Do
interesting.
that. Whatever we've got left.
And here's another one of those boarfish again,
How's that video?
which you've seen a
That's
couple of
nice.
today.
Even geology-wise, this is a lot less going on.
Yeah.
Notice that there's a lot less fissures.
Left and right,
There
the
are a
vehicle's
lot less
wider
broken
now.
rocks. This is a much more intact landscape.
We could swing the upper swing arms forward. The starboard upper
It's
might
interesting,
be.
that boarfish seems to be a little bit intermediate in color between the really yellow one that we saw earlier and then the ones with the reddish stripe. That one had a darker stripe, it wasn't red, but
Up might be
I was
down,
maybe there's
out.
some individual variability or behavioral control over that,
Is that
the color
sometimes
pattern.
depending on whether they're started or not.
I think
I think it has to do with how many cocktails they've had, potential liver disease maybe, I'm not sure.
Okay, I would stay with that.
so. It would only
Are you
be
insinuating that
swinging the
fish had
already
jaundice?
rotated as far to look as far forward. So it'd only be swinging.
That's what I was
the arm itself
insinuating.
More forward.
Yeah, I wouldn't want that. We've got plenty of light in the
Okay.
center.
For those of you who are listening
Pilot,
on the
is
line
that
who are
just
not
going
part
up
of the
the bottom?
shore-based science team, that fish probably doesn't have
Like a little-
jaundice.
with the dark spot in the center.
Oh, what?
So it's only be on your right. As you
But do you
back
think
up.
it still drinks heavily?
Here comes the swing.
That little mound?
Seawater, possibly. Wait,
Maybe
it doesn't drink?
Do fish drink?
I think they
To the
have
right.
to ingest some form of fluid.
Water, yeah.
Bruce, could you please enlighten us on the mechanisms of
I'm
fish
gonna spin
drinking?
down the pack then.
Come in. Come on in.
I'm going to pass on this. I've got a fish physiologist, so I'll step back from this one.
it's too perfect.
There's also something living behind it.
Tilt down a little more.
I don't know.
Ooh, what's that weird mound? What's caught your eye, pilot?
Oh, actually, it caught video's eye. I'm just trying to get a good look at it. It's this little bump.
Someone want to tell us about this little bump on screen and why we're looking at it?
We're looking at it because it was eye-catching, and I can't tell you what it is.
I think it's an unspecified animal.
Nav, if you could start that again. Nav, if you could give us an update as to how
Yes,
far we have left
exactly
to
this.
go till the waypoint, please. That sounds absolutely great. Thank you.
Got a crinoid hanging on for the ride. Let's go.
Fish on the left edge of the screen. Come to the
Other
bottom.
fish on the left, I think. I think it might be a fish.
Yeah.
Looks like another one of these
Bottom of the screen
Kochikzi,
just went
the thing about headed.
I just got a little momentum. I gotta stop. I saw it. Come on in. See this guy?
Oh, yeah, another one of these fish we've seen before.
This is the chickpeas, looking with headed. I gave the wrong genus when I first saw this, but and that name is based on photographs of a species from Japan. I don't know if it's reported from Marianas or not. I'll have to check.
Thank you, Bruce.
Video's clear.
Alright, let's continue on then.
Do we still have a crinoid attached to the ROV?
There is one crawling around on the bottom of the wing, yeah. Which actually is going to make landing next time interesting, because he's right where I usually plant to land. I don't know if I can see him still. See a little bit of him. I could see him really well in my cam. And he's crawling up. Sure. It appears. I'll throw them in the camera up here real quick. Co-pilot.
Sure.
So actually you can see it. You see that?
So we definitely have a stowaway on board. On the under the starboard skids of the ROV, there's a Comanche crinoid which has like jumped on for the ride.
He was hanging onto the front, and then he moved around towards the bottom, and then now he's coming up that side.
I know. So Betso, he's gonna make it all the way.
We'll see.
I think maybe he's on the. He wants that rock back out of the geo box.
Okay. Again, just this is such a contrast to the feature we were on yesterday.
I'm not entirely sure that's true. I think if we had kept going yesterday
It may have turned
and
into
gotten all the
this.
way up to the top, this is what it would have looked like. But we were moving up a different part of a slope. I don't think we ever made it there. I think it definitely shares a background
Yeah.
with yesterday's dive.
I think that's a pretty fair assessment.
Crinoid has decided that's his new home.
I had expected this to be a little bit more productive, though, because of the fact that we were doing fisheries and corals dives.
Yeah.
I guess I thought that getting to this top would mean that we'd be teeming with life.
I agree.
Co-pilot.
Say again?
You can go back to the. Oh, you did. Oh, video's got it. Poke that.
So if you're just joining us, we are surveying a
rotate
site
to get you in
called
Yep.
Santa Rosa South, which is just south of Guam. We are currently in an area where we're supposed to be looking for commercial species of fish as well as coral,
Okay, I'm about
although that is not
20
totally apparent
degrees.
from what we're seeing.
30 degrees
Perfect.
starboard of the line.
We're currently at 287 meters
port of
depth,
the line.
and we should be finishing our
Let's get
dive
back on the
within
line.
15 minutes.
Yeah, we're so far over there.
We've
Still 100
collected
right
several
now.
specimens today. 2 rocks and 3 biology
profile.
samples. as well as we currently have a stowaway on our
We're
ROV.
going along at
There's
100,
a Comanche
right?
crinoid attached to the right
Great,
skid
thank you.
of an ROV, and it may very well make it to the surface.
Copy.
Is this on
Okay.
track now?
Yep.
I saw that go.
I think Deb was 100% right in saying that, you know, we had heard that we were going to be surveying these areas where there was going to be potentially commercial fish and commercial coral species, and we sort of accepted this kind of luxurious coral garden, you know, which is most certainly what we saw at the end of our dive yesterday. But this time we are a bit shallower, and we're just not seeing much of anything, really. I'm sure if we were to stop and go closer, there'd be a couple small invertebrates here and there, but just nothing like what we ended with yesterday.
So Ricky Grigg, who is a very well-known coral biologist, he always used to describe what he considered a coral dead zone, sort of between the precious coral depths and when you actually got up to the around 200 meters where a lot of the bottom fish were located. And while we do find coral in that zone, he was sort of remarking on the fact that they just sort of take off. Maybe there's a transition between some deeper ones and some shallower ones.
Thanks for the explanation. That's really good to know.
More blue, less. No, it's not too far. Well, what did we say? We just do like a partial shot. Maybe there's something smaller that we're missing. A little bit.
Also, this is probably not optimal habitat for the bottom fish species that we were looking for. This is quite flat without any type of features, not a whole lot of prey on the bottom. One of the species of bottom fish tends to hang out near the bottom and eat benthic type of animals. The other is a schooling species that's way up in the water column. But I doubt that this particular terrain would be suitable for either one of those.
Yeah, there are not a lot of areas where fish can kind of hide, are there? Shelter away from predators or take refuge.
I'll swing. Do you want to move forward more into the center of the frame?
Right. The one species of Edilus carbunculus, it's kind of known to be more of a solitary species, somewhat territorial, and it needs shelter because it's smaller. It's predated upon by large amberjacks and perhaps by sharks as well.
We see the backside of the ROV all day long.
Oh yeah, so what you're saying is backwards to me. So backwards is what I would call that. Backwards from our normal track.
Yeah, just cut across the screen in that general way. Makes for good edit videos.
So one thing I'm delighted about is we saw the lower limit of the coral, and then we've also fleshed out the upper limit of the bed as one saw transition between corals and sponges. So it's really quite informative from understanding the community.
Come back to do a reverse.
Chris, would you mind repeating all of that again? You were very broken up, and I think it was a very important statement.
Turn around and do it again going the other way?
Yep.
I was saying that one really great thing about today's dive is that it looks like we detected the lower limit of the coral bed, and we also found the upper limit of the bed, so we actually can get a really good estimate of the size, the vertical size of the bed itself, not the lateral size. And we were also able to record some transitions in the community itself. So this was a really, really valuable dive for understanding some of these deep coral communities.
No, that is an extremely important point, and yes, you are very, very right. I'm sure on the other coral and bottom fish targets, I think there are only one or two more. Is it one or two more?
That's
Zealandia,
good stuff.
yeah.
Got that early in
I'm
the
sure
expedition,
hopefully
so
we'll
we
be
can
able
use that
to
throughout.
gain that kind of information as well.
Go up to the upper corner and come down to the bottom right corner. As long as we keep making tracks and keep looking for new things. What is that rock thing on the right, the center?
Looks like a rock thing.
So if we had a few minutes, it might be good to just take a shot at one of these crinoids just to make sure we've got this one documented, since it seems to be the only thing around at these depths.
Yeah.
Minus 56.
Fish.
Did you get that, pilot?
I did, yep.
Great,
Sorry.
thank you. No, just checking. These ones do look a little different
Bruce
to what
was
we've seen
interested in
before.
that fish. He was just zooming in. If he could zoom in again.
They want to see that.
Did you see a fish
rock
there, pilot?
Fish.
Well, we've actually got ourselves in a... We were doing a beauty shot since there wasn't much to look at for Rome.
No, I didn't think.
We're
I
out
didn't see a
here.
fish.
There was a small fish, but we're out here where we really should catch up to my co-pilot.
It's down below the camera now, and not sure.
It's right there on the right leg.
There he
Kind of
is.
sitting on the bottom.
There it
It's
is.
on the right on the basket edge.
And there's also a crino
It just
on your
moved,
left
stopped
skid.
moving.
Yeah, he's been living there for a
You guys
while.
have great
Left.
eyes. I still have not seen this fish.
Oh, on the ground, yeah.
It's center right, almost exactly center on the right.
Oh yeah, there we go.
Let's start
Yep.
to come in.
Never would have seen that.
Absolutely never would have seen that.
I guess it's great we're both watch leads, right?
That's why we have people on
Is
shore.
that how you count? to
Exactly.
14?
The value of
That's how I count
telepresence.
That's how I count to catch up to Syria.
Lots of people to see things that we may not, as well as lots of people to ID things that we can't.
Yeah, you can just shoot straight through there.
So,
That's a good image of the fish. It looked like it could have been a Putvanthia species, but a different one than the one that we were seeing deeper. It had more of a blotch pattern, strong bars pattern.
We'll get a shot of that one of the crinoid. I'm just going to get a little bit safer spot.
yep, that's okay. And the good news is we are nearly to our end waypoint.
Absolutely. Come on up.
Yeah, all in right now.
Let's get this down to the next one to see.
That was a big heave.
This might be the winner here, Roland. You want to take a quick look at this one?
Oh, yep, get as close to it as you can.
All right, come on in.
So Chris had to step out of the room for a moment. He has a telephone call, and so he's not here to see the crinoid that he asked you to zoom in on. Thanks.
That's okay. We're going to take a minute or two here and look at this crinoid. I'm going to start going through our guide and